Well, I think you can hear us. And first of all, good afternoon, good morning, good evening to wherever you are. And thank you so much for joining us to our very first session of Meet My Lab. We are all new to this, so please forgive if there are minor IT hiccups, and we had to change the platform in between just to make sure that we all have a successful session today. So it's strange not seeing anyone, but I think we're going to have an excellent session ahead of us. My name is Susanne Renzo-Vasu, and I am representing the EURAXESS worldwide project here in Southeast Asia. I'm based in Singapore, and I can tell you we have such an exciting panel ahead of us. I'm really looking forward to this. Meet My Lab was born out of the necessity that we currently cannot meet each other. But of course, as we know, the problems are even bigger than they were before the COVID crisis. So the need for us to work together is stronger than ever. So what we wanted to do with this platform is to help researchers, especially young career researchers, to meet each other online, talk about their research, find common ground, and hopefully walk away with partnerships in the making. And what we're focusing on today, of course, is vaccine research. I'm not going to say a lot more about this because I have a fabulous moderator who's sitting next to me, and I'm going to introduce them in a second. I just wanted to just briefly introduce you to EURAXESS, which is the organization that I was just talking to you about. EURAXESS is a European Commission funded project, and it's dedicated to supporting mobile researchers. So what we're doing is really trying to eradicate the barriers that make it difficult for you to move around and to work with each other. It's a project that is supported by the European Commission, and more importantly, it is supported by 36 European countries. And what they do is they basically put all this information that you as young researchers need in one location so that you can plan your next career step. For example, funding opportunities, job opportunities, fellowships all across Europe. We have a partnering tool, which is a little bit like what we're doing today, an opportunity to reach out to other researchers across Europe, across the world, to build research networks. We have a hosting database, which is a platform where you will find advertisements from supervisors and universities in Europe and across the world that are looking for young researchers to spend some time at their institution. And of course, there's lots of information there. What's it like working in Europe? Where are the funding opportunities? And here under our personalized assistance platform, there are real people that are waiting for you to contact them and to help you with making your mobility experience a reality. I am part of the so-called EURAXESS worldwide network. This is a number of offices that we have established across the globe to make it easier to link up Europe with our main research collaborators. I am based in Singapore in Southeast Asia, and you can always send me an email or click on this wonderful QR code that I have created to find out what other activities we are up to. For example, we have lots of workshops on proposal writing, etc. And of course, this leads us to the Meet My Lab platform, of which we're having the inaugural event today. And of course, I have placed wrongly here my slides, because now I actually want to introduce you. If you could make me stop sharing my slides, how would I think I'll do this. I've stopped, right? Then now it's my turn to introduce you to our moderator for today's session. And it is the young gentleman who's seated here. You can actually see my hand in the screen. It's Weiji Po, or Po Weiji. He is currently the Global Project Manager at Healand in Singapore. And he's also active in the Boston and Singapore biotech ecosystem. Prior to the job that Weiji is currently holding, he was an entrepreneur in residence at the Center for Technology and Development at Duke-NUS Medical School in Singapore, where he was focusing on commercializing vaccine technology. So obviously he's the right man for the job today. Weiji holds a PhD from the US, from Johns Hopkins University, where he also co-founded a universal HPV vaccine startup, and he also completed postdoc research at the Dana-Farber Cancer Research Institute in Boston. So he is really the man to lead the session today, and I will very gratefully hand over to him to take the floor. Thank you, Weiji.
Thank you, Suzanne. So, hi everyone, and thank you all for calling, for listening in today to this webinar. Can you guys hear me? All right. Yes. Okay. Good. All right. So, so I see that we have about close to 30 attendees today, and I think it's really in line. Hopefully, as more come in, really to echo what Suzanne is saying, this is meant to be a small tech niche sort of gathering networking event for folks that are interested in vaccines. And I've been very privileged to be invited by Suzanne and EURAXESS to moderate this seminar, this webinar, and also to have the privilege of having three very well-traveled, young, exceptional researchers to share with us about their experiences working with industry in advancing two of the close to 30 COVID-19 vaccines that are currently in clinical development. And when I was given this opportunity, I think it's really great for us to hear from the PhDs, the researchers on the front lines. You know, we've heard a ton of it from the PIs and the regulators, the government. Now, I think, you know, learning what the experience is at the front lines of doing the bench research, as well as providing the opportunities to interact and potentially collaborate with these two great institutions, Duke-NUS Medical School in Singapore and Institut Pasteur Institute in France. The three ladies have generously allowed me to share their contact information here. So if there's anyone here who's interested in working with them, now after listening to the talks, feel free to reach out to them at the following email addresses. And you know, that's what we like to see. We like to see this whole collaboration going forward. So without, I guess this goes without saying, COVID-19 is one of the biggest, one of the most near-term pandemic that's hit us in human history. And with the advantage of having IT and information sharing, we're actually being able to have near-time insights into how the pandemic is evolving. We know that, you know, since it was first reported just 2 weeks ago, the worldwide cases for COVID-19 has exceeded 30 million cases. In terms of the deaths that's been attributed to COVID-19, it will be close to 1 million in a couple of weeks. So it's definitely something that impacts not just a couple of countries, but every single country in the world, and with severe economic impacts. And that's where we're seeing a lot of public interest, not just from the pharmaceutical companies, but also from the public, from regulators, from governments. And so this is just a snapshot of the data I've gotten from an article back in August, but I'm sure the numbers will have jumped by now. That's been for us, and I hope I can still consider myself part of this ecosystem. For us in the vaccine development space, for the longest time, it's always been something that's been seen as long time to market, not worth the investment, and there's only a few couple of pharma companies that are working on it. But with COVID-19, given the dire impact on economic and travel, on all aspects of economy, we can see the governments of the world coming together and the pharmaceutical companies coming towards the world, the research institutions coming into starting to accelerate all this and work towards developing vaccines. And I think this is a really, really exciting time for us to, as a community, to not just advance advance our understanding of how vaccines works, vaccine development, vaccine preclinical and clinical development works, but also using platforms like this, understand, how the different platforms different from each other, what are the considerations, how do pharma, for example, think about this when they seek partnerships. So with the increased public interest, obviously there's a lot more, we're seeing a lot more, with great power comes great responsibility. And I think that with the pharma industry in general, they're starting to adopt more open policies. And just a week ago, the major pharmaceutical companies have come out to just release the blueprints of how they're conducting their clinical trials. And I think that going forward, this is something that is nothing but beneficial and good for us, for the industry in general. And I think just to overcome and to add more credibility to how vaccine development works, because very often for most people, it's always a black box. It's something that takes 10 years, and then we have no idea what happens after that. And so today I am very privileged to have researchers from two institutions working on two slightly different modalities, but both are technologies that are already in a phase 1 clinical trial, human trial. So with Duke-NUS, we have this RNA vaccine from Efficacious Therapeutics, similar to what we see in Moderna. And with GB and her work on coronavirus, they're using a measles vector vaccine. So this is just two of the many platforms that are being investigated right now in different forms by different entities in clinical trials. So without further ado, I think we will hand over the slides over to the speakers. And then how we'll do is we'll first let Eugenia and Rukie speak about their work that they do at Duke-NUS, show us a bit about their lab. As you can see, that's where they're sitting right now. And then we just hand over to GB and for her to do the same. And then we just have a nice panel discussion. As they are talking, feel free to ask your questions in the Q&A session, in the Q&A box that you see at the bottom of your screen. And then we will just ask them towards the last stretch of the session. So without further ado, I would hand over this to Eugenia.
So, yeah. All right. So, good afternoon, everyone, or morning, depending on where you are located in the world right now. I think first of all, we really like to thank Suzanne as well as Wei Jie for. Is the volume okay?
Is it not loud enough?
Okay. Okay. So yeah. As I was saying, we really like to thank Suzanne as well as Wei Jie for really giving us this opportunity today to share more about the work that we're currently doing at the Bioresearch and Experimental Medicine Center at SingHealth Duke-NUS, or Viremix for short, because that's a whole mouthful. So maybe a little bit of background about our lab and how we started. So we were established in 2017 with a generous donation from the Tanuto Foundation. And I think one of the main goals at that time as to why the lab was established, it was to try and aim to accelerate clinical translation of vaccines and therapeutics by working with both academic as well as industry partners to be able to develop a suite of immunology tools as well as molecular tools that can allow us to very accurately assess both safety and efficacy of the vaccine and therapeutic candidates that are in the early clinical development phase. So if you refer to the diagram on the left, this is where we're situated right now. So we are jointly held under both Duke-NUS Medical School as well as SingHealth. So SingHealth is one of the largest public health clusters in Singapore. Where we're situated is actually right next to Singapore General Hospital, which is also the largest public hospital in Singapore. You'll also see that we have a very nice link with the investigational medicine unit at SingHealth, which is an early phase clinical trials facility that is we collaborate a lot with in terms of these early phase clinical trials that come our way. And so some of these projects have included both vaccines and therapeutics against SARS-CoV-2, yellow fever virus, chikungunya, dengue virus, and so on. And I think that's the space that we are at right now, and we are always actively looking for collaborations as well as research partnerships with both academic as well as industry partners.
Okay, so this ecosystem that Eugenia just described, Viremics was set up to take advantage of it. And the way we sort of, one of the things that we are interested in doing is using both old and new technologies. So definitely for vaccine development and to accelerate vaccine development, we wanted to use new technologies like the omics platforms. So what we do is we have a lot of these gene expressions or molecular tools, metabolomics, cytokine expression. What we do here is we integrate all those omics data and we try to use that information to accelerate vaccine development. Now, on the other side, we also have the tools that are more traditional tools that are being used for regulatory purposes to license vaccines. So basically we have tools to look at antibody responses, neutralizing responses if it's a virus, also to look at T-cell responses. So these protective immune responses, we have those assays set up as well. And another thing that falls under omics platform is we also have what's known as system serology. So with serology, we also go an extra length and do look at antibody effector functions. So we're trying to really use our ecosystem where we're connected to the hospital, to the university, where we can set up clinical trials and also put some new technology to look at how vaccines are developed and whether we can use these new technologies along with the traditional technologies to accelerate vaccine development. And that's really what Viremics was set up. And we were set up, like Eugenia mentioned, about 2.5 to 3 years ago for this very reason. If there is an outbreak, a pandemic, and we need to accelerate a vaccine, then we are there to do it. So this is sort of, and I think we also need to highlight that we are starting with standardized testing. So we do have ISO certification for some of our molecular testing.
So I think as with many other labs around the world, some of the challenges that we really face during a pandemic lockdown. So in Singapore, we did not exactly go into a complete lockdown per se, but the Singapore government at that time introduced what was called a circuit breaker phase. So it was meant to break the chain of transmission and hopefully try to bring the numbers of cases down. And so in Singapore, we implemented this circuit breaker phase from the 7th of April. And even until now, certain restrictions remain in place, but a lot of other restrictions have been lifted slowly in places as well. So we did continue our work in the lab on vaccine development during the circuit breaker phase. We had special permission to work in the lab, so we were considered an essential service. So if you look on the right-hand side, this is actually a public messaging from the Ministry of Trade and Industry in Singapore on the types of essential services that were actually exempted from suspension. And we actually fall under the category of the biomedical sciences right at the corner over here. So during the circuit breaker phase, of course, with that permission that we had to continue working in the lab, we did also have to implement certain measures that would actually help in potentially preventing any infection from occurring. And so some of the measures that we put in place were, for example, things like working in split teams that were non-overlapping, and also we had limited time in the lab. So for example, we had one team that would come in the morning and leave before the other team came in the afternoon, right? So in addition, I think what a lot of other labs face as well, and it's a problem that we face as well, was that there were global supply limitation of reagents and consumables. So, you know, we had to plan ahead and try and make sure that we got as much resources as we had or as we could have had at that time. And also with the lockdown in place in many other countries, there were also delays of reagents for certain experiments, certain reagents that were typically used for molecular work. There were also like shortages faced throughout the world, and so we just had to work around that and make sure that we had proper planning in place and to ensure that that work could actually continue as if it was normal times. So these were some of the challenges that we faced, and I think now we're just gonna bring kind of like everyone on a short, like maybe tour around the lab. So follow us.
Okay, better.
So basically, this is where we're situated, our lab. We don't really take up too much space, but where we're in right now, this is the molecular corridor. And so this is where the bulk of the molecular work that we were talking about earlier, that's where it's all done. So I think similar to many other labs, we have the typical lab equipment that you see, fire safety cabinet, we have real-time PCR cyclers as well as cyclers. What's nice is that we also have the automated extractor, which helps a lot when we are extracting high quantities of samples at any one time. We also have the nano string sprint in the lab, which has been very instrumental in allowing us to do digital profiling of gene expression that we are getting from vaccine trials as well as other therapeutic trials that are being carried out. And it's really given us like a hold on assessing both safety as well as efficacy of candidates that are currently in early phase clinical trials.
And here we also have more lab space here, and we do a lot of the serology, and a lot of them are fluorescent based. So we usually have the lights turned down or turned off, and we have some of our colleagues here running some experiments, both for vaccine trial, but also other COVID trials as well. And again, it's set up as a very typical serology lab. So again, PS electron readers. We also have equipment in other common spaces as well, like the next equipment, where we do cytokines, we also do serology, and also a lot of the omics happens in core facilities, which is again in restricted common spaces. So we can see down there, it's an open lab concept. So we have other colleagues which are hospital-based or clinical-based. So we're really in a setting where we mix academic scientists along with individuals who are doing very clinical work. So this is sort of where we're set up. Okay. That is sort of a tour. We wish we could have given you more of a tour, but I think some of the spaces are restricted and we're not allowed to take, videos of it. But this is our little space here where we can show you guys. So yeah.
All right, thank you, Rukie. Thank you, Eugenia, for showing us your lab and explain to us your work. So I'm a little bit biased because I'm based in Singapore, and I would say the lab where they're standing, it's probably less than 300 meters from the clinical trial unit. So when you want clinical samples, you just get it straight from the lab, and I think one of them just runs over with samples to the lab. So I think it's a really interesting setup. And that's really where they're positioning the lab is to support the early stage clinical development of vaccines. So now we can move on to GB.
Hello, I'm still here.
Yes.
Hi. So, yes, for our lab, first of all, I would like to thank EURAXESS, because I'm also, I'm originally a researcher in the institute in Thailand called NASDA, and in the biotech, and then it's because of EURAXESS connecting us with Institut Pasteur, and we have been collaborating for I don't know how many years. Since 2013, I came here to visit the lab here at Pasteur in 2013, and since then we've been getting grant, getting funding, and then keep coming back to collaborating together. So my original project is on the coronavirus. It's a porcine epidemic diarrhea virus, and which is this originally a problem in Thailand, in Asia. And then I came to Pasteur with the problem, and we were figuring out what we can do together to combine our knowledge in PDV, which is the coronavirus, and the platform for vaccine at Institut Pasteur. So right now our lab is under Dr. Frederick Tongji. Our lab is called Vaccine Innovation Lab. I'm not sure, because sometimes in French they're like innovation vaccin innovation, but it's a new lab, because actually my boss, he's the previous lab is the genomic viral, has to close down because he's supposedly to be retired, but because his work is still going on, so we continue as the innovation vaccine. So this lab is based on measles live attenuated vaccine. He's developed this infectious clone, or a vaccine recombinant that based on this live attenuated vaccine. This vaccine has been first implemented in the 60s, and this has been passages, it's safe, and it's from the wild type pathogenic strain that passage until it's attenuated, that not cause the disease in human. So this is one of the most safe, and I think most of people had measles vaccine, except the people that, now it's another issue with the anti-vaxxer. So next slide, please. Yes, and our platform is based on plug and play system, because we create, actually my boss, he developed this by that we could add additional gene into the measles vector, and then we can, the measles can express any heterologous or the additional genes of other viruses. And one thing of the measles too is that because of there's also established manufacturer, so if there's any emerging diseases, and we have all these manufacturers ready to implement our vaccine candidate. And also one thing, one benefit thing about measles vector is that the measles virus is in a pleomorphic form, which means it's flexible. The virus encapsulation is very flexible, so it could, we could add extra genomic DNA into the vector, and it can still express, because some of the vector is limited to the size of antigen that we can add in, but measles, now we can add up to antigen up to 7 to 8 kb. So it's very beneficial. Also, this vector has been in several preclinical trial. We have chikungunya now, that is the most advanced of our vaccine candidate. Now it's in the phase 3. This measles chikungunya been how can I say this? It's been actually Demis is the one who bought the license for the measles chick, and then they've been testing in the phase 1 to phase 3 trial. And then with this collaboration, when SARS is become pandemic, CEPI approached us to lead the program to produce measles vector vaccine candidate. So, and these are the collaboration between Institut Pasteur, Demis, and University of Pittsburgh. As we can see, there's several vaccine in clinical trial, preclinical trial, and then there's Zika and Lassa that's also entered the phase 1 clinical trial too. But the interesting thing here is that measles chick has been developed for 8 years, and now we just reach phase 3, which is bring us to SARS, to that. It's amazing. If you can move to the next slide. I put this into this slide that the traditional vaccine development takes years to develop, because first we need to design the antigen, design the vaccine, and then it's enter the preclinical trial, which is in mouse model, in monkey model, and then once the preclinical trial is giving a good result, then we start producing a small scale to be safe for human, and then go to human phase 1 trial. And then actually each phase is need to be wait to see the result. But now with the pandemic of the COVID, it's interesting that everything is mushed together. It just, we have to thank the company, to the industry, because they take risks. This is a big risk for them, because they manufacturing the candidate along with the test in the preclinical trial, clinical trial. So they're taking a big risk, because usually they would not run this until we have the solid result. So, but this will increase the process of vaccine development like tremendously. As I mentioned earlier, that measles chick has been taking 8 years to enter phase 3, but for this COVID vaccine, you see that it's already entering the phase 3 clinical trial, which is amazing. And I mean, for us, working during the confinement, it's been very interesting because you know, the Europe has been locked down. I think we locked down in March, April, and then we reopened everything in May. But our vaccine research start in February, and which with synthesizing the gene and then creating the vaccine. So it takes, we pretty much working every day during that time. So we have everybody. So it's obvious that I'm in the lab. We have been working pretty much every day, and I show you a picture of me biking to work, because we were not allowed to take any public transport, because in case we need to work, so we should not get sick. We're not allowed to get sick. So that's why I have to bike. I need to buy a bike, bike to work. And then at work, you can see that because a lot of restaurant around was not open. So we have this meal delivered to us, and then you see that we have to go through line in keep the distance. So these are also the group that we've been working tirelessly for 2 months, every day for 2 months. And I think one of the most challenging for the COVID vaccine development is the fake news. The fake news it's crazy because it's kind of deterring the people who work for the vaccine development. So I would like to raise an awareness of this fake news online, that because we're working so hard, we're working maybe 80 hours a week, and then we hear that, oh, the fake news of we are the one who create the virus at the first place. And it's just, and then actually my boss, he got affected the most because of this fake news. Even call him a father of the COVID vaccine. Father of the COVID virus, the SARS-2, which is, it's outlandish. It's just, I cannot believe that someone would call that. And then they even call him, he got a telephone for a threat and stuff at home, which is, this is ridiculous. So I think this is one of the deterring, discouraging for researcher for doing so hard work, and then this is what we got, and then it's, so this is all the stuff that has been happening during the confinement and the vaccine development. It's interesting that it's been 6 months almost, but to me it felt like the time just like woof passed by. So I think Eugenia and the other lab is feel the same too, right? I think. Yes. So, and then I think I should bring everyone to show the lab, right? Okay. So, okay, now this is the technical stuff. But let's see. Hey! Okay. Up. So, hello. Let's go see the lab. Here's the ladies. Who's been working so hard. We've been working every day together. We're eating lunch together, and I think I should put my mask on now that everybody gets to work. Here is our share office, and I'll try to walk slowly. So the lab are, we have this room. The lab is normally like this. Everyone would know that the labs are pretty messy. So, and here's are the place that everybody mostly do cloning, some small stuff, and here is the culture room that we do cell culture. Can you hear me? Hello? Everyone hear me well, right?
Yeah, we can hear you. We can
okay,
hear you.
Thank you. Because like it was quiet, and I'm like, is anyone there? So this is quite an old building. Then I'm not sure if I go through down the hall. There's P2 plus down the hall, but if I go down there, it's like a dead zone for a signal. There's really no signal. So let's say I can point down the hall to the left. Hello. Down the hall to the left, there's the lab. And then, hello, that's my coworker too. That's my colleague. And I think it's probably pretty much the lab that I can show too, because, and then we also have main facility that is like a platform that everyone can use in the main new building. And so I don't know how much I can show, but these are labs, and like we have mice hanging. So yes, this is where we work, and then this is, if like the MTV, they would say like, this is where the magic happened. So I don't know. Okay, maybe we can get to the question. I hope I did all right.
Yeah, thank you, GB, for showing us your lab. So I realize I'm actually gonna send a poll to everyone. I've seen I see that we have about 40 of us here. Just answer, like, let us know what your background is, and I see quite a bit of researchers in the chat channel, but feel free to vote so we get a better sense of what everyone else is coming from. Just gonna give it. All right, just gonna give it a couple more seconds. Just gonna close it. Wow, interesting. Oh, wow. Ooh, wow. Okay, I'm gonna close it in like 5 seconds. 3, 2, 1. Right. So interesting, very interesting that we're seeing quite a bit of folks that are not in vaccines that are they're calling in, they're listening to the conference today, too, from the public, and I hope this is helpful. I think for one of the reasons why we wanted to add this in this lab component is just to get everyone to see what it happens in the academic lab, like letting you know that there's no fancy conspiracy going on, but also just let you know that all across the world the labs are pretty much the same, right? And I think what really matters are the people who are doing the work itself. So on that particular note, I'm gonna use this time to do an introduction of our panel, to actually properly give an introduction of our audience, of our panelists. But also for the audience here, feel free to use the Q&A function and start asking the three researchers any burning questions that you have.
Il est le modérateur.
Le modérateur.
I think GP is speaking French. All right, so yeah, so feel free to use the chat function and talk to them. But in the meantime, I think this is the part I should have done earlier. But let me give you a proper walkthrough of where these three young researchers come from. So we start off with Rukie. So PhD from University of North Carolina, then did her public health, master's in public health from London, then ending up stopping over in Vietnam, right? And before coming over to Singapore. So extremely well traveled. And if there's one word I can use to define her research and her work, it's really systems serology. And we can ask her that during the panel discussion on what exactly that means. So obviously with her and Eugenia, the platform that we're working on is the self-replicating microRNA COVID-19 vaccine from Extractor Therapeutics that started phase 1 trials here in Singapore in August, in the middle of August. So Rukie is also fairly active in science communication. So had a couple of articles in our Singapore newspapers talking about also that what is COVID, there's also this other big virus going on, which is the dengue virus infections, which actually is peaking this year compared to previous years. So next we have Eugenia, did a PhD at Duke-NUS Medical School in Singapore before joining Viremics. And her role is really more of the ISO certification for Viremics, the lab itself, the 2 bench spaces that you're seeing behind her, as well as really, I think this is really the exciting part, really working with Rukie to really think about interesting ways of doing clinical trials that allows you to speed up clinical development you know, and I think that for the longest time when it comes to vaccines, we've been relying on endpoints like, you know, self-reported symptoms that may not fully recapitulate the molecular understandings. And I think this group is doing a great job at trying to define and putting science into regulatory action. So Eugenia also has been featured very widely on our social media in Singapore. The only one who doesn't get a photo shoot out of this four outstanding, three other outstanding CEOs of big companies here in Singapore. So then we have GB. So again, another very well-traveled researcher from Thailand, went to US, came back to Thailand, and then went to France to do the research. And as she has explained, her focus has been on the measles platform, first targeting porcine coronavirus, and it's like the speed at which that transition into COVID-19, and how that just goes to show how adaptable the platform is. I mean, from a personal point of view, the company that I used to have was a virus-like particle, and anything more than 2 kb, we're like, that's not going to happen. So when you mentioned, the moment you mentioned 7 kb, that's like a dream come true for vaccine development. Yeah. So yeah, and for she is she's also very good on science communication. Like if you go on YouTube, she has a rap video talking about her work. If you want to find out what it is, very simple. Just search "Pigs Don't Cry" on YouTube. That's the first result you get. I don't think there are that many videos with that title. Yeah. So yeah, with that, I'm gonna stop sharing and actually going to do another poll if I can find a way how to do it. Here we go. So when do you expect the COVID-19 vaccines to be widely available? So after that, after we get the results, and then we'll just go around between the two groups, you know, just to understand a bit more about. I think there are a lot of researchers amongst us here, so maybe talking about how the experience was, you know, working with your respective industry partners, as much as you can share with us, and what was new to you, what was unexpected, and you know, any advice for other researchers out there who are thinking about the same thing. Like, you know, I think I have this awesome platform technology in my lab. How do I find people how do I get companies to come and look for me to collaborate? All right, so give me a couple more seconds. Okay, so here are the results. Quite a fair, even spread of optimists and pessimists. All right, yeah, I think what we can all agree on is we cannot wait for the vaccine to be available so we can get back to the normal, right? Okay, great. Thank you all for participating in the poll. So let's just open up to, so we have about 10 more minutes to go. I think let's just open up to, let me look at the, there's a lot of interest, there's a lot of questions from, okay, I'm gonna pose this question to the panelists and answer this, and then we'll go into the Q&A that the audience is asking. So tell us about how it was, you know, working for GB, how it was working with Thermis, you know, and how that's changed, you know, now that Thermis got bought out by Merck, and Merck's the one advancing it into clinical trials. And for Eugenia and Rukie, you know, how is it like working with Extractor Therapeutics? How does that compare to typical academic collaborations? So we'll start with you first, GB.
Okay. So first of all, I mean, we've been working with Thermis for a while, but never in this urgency process before. It would, like, you want to know like the process we work with them, or how they connecting with us. It's just, first they were approaching us for buying the license of the chikungunya vaccine, and then since then we were developing together the clinical trial for the chikungunya, and then for Zika, and now for Lassa. But for the COVID, everything is in the super speed. Because during we do a preclinical trial, they also recruiting the preparing the clinical trial already while we starting the preclinical trial. So, and everything is going to the speed of light. So they asked for the seed vaccine in May, that we need to have the seed vaccine in May. And then we start making the virus, cloning the virus in early March. So March to May, that was tight. So, and then for virus, it's not like we can just grow. We need to do the process and let them grow in their own speed. So if they want to grow, they grow. If they don't want to grow, they just give up or something. But it's just it's too fast for us.
Do you think that's going to be the norm going forward? Is that now like the benchmark for everyone in your lab, that we're going to go from concept to virus to a vaccine within 2 months? Like, is that the new norm for all your PhD students and the postdocs?
I hope not. And unless we have I mean, it could be done, unless but we have to have a 2 team that can do the same thing. Because like this team can go all out and then switch to the second team to go all out, then that works. But if we have just one team to go all out like this all the time, no.
All right, thank you, GB. All right, so now over to you, to Rukie and Eugenia. So how was it like working with Infectious? Whatever you can tell us, and what was the interesting insights that you gained from this process?
So it's actually been quite a fun process, definitely stressful, but we started, I think when Enyong told me about this vaccine study, he was like, I mentioned, hey, I'm interested in working on COVID. He said, sure, you want to work on vaccines? That was February. He's like, yeah, we're talking to COVID, come. So basically, since that day, it's been nonstop. We've just been working. But it's been interesting because, so we worked with companies before. So we work with industry partners quite a bit. But definitely there are 2 things against us in this pandemic, right? Both knowledge and time. So knowledge and time. And what's nice is the actuaries actually, they understand that we don't have all the knowledge. So we might set up an assay, we might do things a certain way. And as we go on, we get new information because there's a ton of research being done on COVID. And then we realize, we should look at this as well, or we should do this instead. So it was the same for the vaccine design, because we started with quite a few constructs and we narrowed down. And as information poured in, we use that information too. So it was a very nice collaboration in terms of, you have emails flying around and saying, hey, there's an interesting paper. Hey, did you see this? And that's also from the company side, not just the academic side. So we saw a lot of that. And I think as we got closer to the clinical trials, just as Gibi said, and I'm sure Eugenia can also confirm this, we had preclinical ongoing and tox studies ongoing, and then non-human primate studies ongoing, and then bam, clinical trials started. It's all staggering, and they all haven't ended yet. So we're sort of drowning in work, but there's a really nice understanding on the company side. I know Eugenia has she basically coordinates a lot of the trial with I think she has one of the hardest parts because she works with the IMU and Health and SGH Hospital to really coordinate that stuff. For me, it's easier because I coordinate the lab stuff and the logistics on the lab side. So that's not, I think she has the harder part. But again, for me personally, it's been a nice experience working with the company. Just that time, we just don't have that time element. Sometimes they get very enthusiastic and they want something. And you're like, nope, we just got the mic. They have to be immunized for 1 month. So, things like that. But yeah.
And yeah, I would like to add too that so I use the mic on the phone so so it doesn't pick up all the voices around. I would like to add to that another thing is that thing is going so fast because the competition as well. And I mean, the science competition is one thing, but with the industry competition, it's very fierce, I think, because there's a lot of money involved because they, like I mentioned earlier, that they take risks to make a large amount of vaccine candidate, then which they don't know if this would be approved and be sold later on, but it's also nice that a lot of government has funding to the company too, so they are more daring to take this risk.
Interesting. Great.
Yeah, so I guess if I can also just add on very quickly, I think one of the largest learning experiences that I've had, working in such an accelerated phase. So like Rukie mentioned just now, we've had experience working with other industry partners, but I think because of the way that the pandemic has turned out to be, it's unprecedented in terms of the speed at which research is ongoing, at which all these various collaborations are being set up. And I think what a lot of people fail to, or at least what we think about most of the time is the actual research that's ongoing. But a lot of times I feel that when actually the actual research starts, that's when it's slightly easier. I felt that the more challenging phase for us was when it's really getting all these studies started up. So it's things like we have to get paperwork in place, agreements have to come in place, and these are all happening at an accelerated phase, and people are working from home. so all these challenges kind of add up as well. And I think with the preclinical work as well as the clinical work and them being ongoing all at the same time, I think what has been very useful is also this idea of being able to conduct clinical trials in an adaptive manner. So we've found that to be very useful, and in our experience working with Arcturus Therapeutics as well as with other industry partners, the clinicians at Singapore General Hospital, as well as our PI, Dr. Jenny Loh, she's had a lot of experience working with adaptive clinical trials, and I think that really helps to accelerate the way that the clinical development is ongoing. So we at the same time in preclinical phase, we were testing multiple candidates and deciding, which one was the best candidate that we would bring forth into the phase 1 clinical trial. And then when phase 1 started, then it's testing different types of dosing regimens, different doses, and trying to make sense of, what is the best way forward. And this is always an ongoing discussion with both the company as well as with the lab, because in the lab, we are actively, generating the data to support further clinical development of that candidate. And I think that's really been the most eye-opening experience for me.
And for the first time, we actually see this balance between reactogenicity and immunogenicity, right? So with any vaccine, you want to balance the two. So it's been quite interesting working with the clinician. And as Eugenia said, adaptive clinical trials is amazing during pandemic times, but in the lab, it's horrendous because
and it's gone.
And it's gone.
You throw that plan out the window.
Throw it because the clinical trial is
just changed
according to,
according to like the data that we just gotten. So
So it's good and bad at the same time. Overall, it's good for the acceleration process.
Awesome, great. I know I realized about where's the time, and so I'm gonna ask GB any advice that you have for academic labs, researchers out there thinking about finding collaborators, and then I'll run into one question that I think is really interesting. I'll ask both of you after. So GB first. Tips and advice for researchers. What do you think about, what do you think is unique about your lab that made companies like Thermis, come to you and say, hey, we are interested in seeing more about your platform?
I think we need to have very good data, very good platform that can be versatile. If you could find a good platform that can be applied to in several emerging diseases, that's for the vaccine. I mean, it would be interesting for the company. And also, I think with the institute, you can also make into a deal that your platform can be sell partly, not just, you don't have to sell the whole platform to a certain company. You can be licensing to just part of your platform. Like Themis only have the Chick and Zika and then SARS too and the Lassa, but the rest and HIV and the other are owned by a different company as well. So if you have the versatile platform, you are golden, I think.
Platform play, right? And I think it's really one of the advantages of like the vector platforms, adenovirus, measles, as well as the nucleic acids, right? The messenger RNA. It's astounding how quickly mRNA vaccines have just taken over COVID-19 development, given the fact that prior to this, there's never been a mRNA vaccine that has been approved. So I think it's on all fronts. I think if you get a regulator here to speak, he or she will be saying, "Oh, this is insane. That's the amount of work, the turnaround that needs to happen. It's almost like warp speed, right? Like what has happening in the US." And I think it's a good sign. I feel like for the first time, people are actually gonna actually think about a more informed approach to accelerate the development of vaccines. And I think part of it is driven by science. And I think that's important, and that's important. That's where the academic research comes into play to say, given what we've learned over the past decade, the century of vaccines development, this is what we think is happening. And like to your point, even about the measles vaccine, right? We don't no longer have to wait for someone to passage a virus for 150 passages in order to get to a safe vaccine. And I think that's really testament to how fast vaccine development has come. And I think this do not waste a good crisis. And I think this is one where the bright light is that we're actually seeing a lot of progress on that front. Yeah. So
I agree that the SARS-2 really gave a stage to science, because the money's coming in, and like everything in science need money. And I think like you said, it's a good crisis in a way.
Yeah. All right. So I'm putting out this slide, and I know we had quite a few questions about technical parts, and I directed to GB. So feel free to email her with all your questions. And there was one question about the human challenge trials that I would love to pick your brains on, but I know we are short of time. So with that, I'd like to thank Suzanne, EURAXESS, and everyone here in the audience for your patient time, in the past hour with us and with Rukie, GB, and Eugenia to share their experiences. I hope this is a helpful platform. And again, if there's anything, if you feel like there are platforms that are of interest, you're looking for collaborations, their email addresses are here. Feel free to reach out to them. And if anything, check us out on the EURAXESS Facebook page. for more of such events coming up in the future. So with that said, I will call this to a close and thank you all for your time. Bye-bye.
Bye.
Thank you.
Bye.
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 Bye. Thank you.